Double-strand break repair. Double-strand DNA breaks or DSBs are most biologically hazardous types of DNA damage and are common events in eukaryotic cells. There are two major pathways for repairing them: homologous recombination and non-homologous DNA end joining or NHEJ. These two repair systems differ in their requirement for a homologous template DNA and in the fidelity of DSB repair. HR directed repair is largely an error-free mechanism as it utilizes the genetic information contained in the undamaged sister chromatid as a template. In contrast, non-homologous end joining is normally error-prone and involves elimination of DSBs by direct ligation of the broken ends. The HR pathway is activated when the cell is in late S or G2 phase and the template has recently been duplicated. This mechanism requires the presence of an identical or nearly identical sequence linked to the damaged DNA region via the centromere for use as a repair template. Double-stranded breaks repaired by this mechanism are usually caused by the replication machinery attempting to synthesize across a single strand break or unrepaired lesion, resulting in the collapse of the replication fork. Non-homologous end joining or NHEJ is used at other points of the cell cycle when sister chromatids are not available for use as HR templates. When these breaks occur, the cell would not have replicated the region of DNA that contains the break. So, unlike the HR pathway, there is no corresponding template strand available. In non-homologous end joining, the co-heterodimeric protein positions the two ends of the broken DNA strands for repair without an available template, losing sequence information in the process. Multiple enzymes are involved in the rejoining process, including DNA ligase 4, XRCC4 and DNA dependent protein kinase or DNA PK. Non-homologous end joining is inherently mutagenic as it relies on chance pairings called microhomologies between the single-stranded tails of the two DNA fragments to be joined. In higher eukaryotes, DNA PK is required for non-homologous end joining repair both via the primary mechanism and via an alternative backup mechanism NHEJ. NHEJ is critical not only for the repair of pathological DSBs as in chromosomal translocations, but also for the repair of physiological DSBs creating during VDJ recombination and class switch recombination. Therefore, patients lacking normal non-homologous end joining are not only sensitive to ionizing radiations, but also severely immunodeficient. When double-strand breaks arise in any organism, prokaryotic or eukaryotic, there are two major categories of DNA repair that can restore the duplex structure. In this image, you can see the physiological and pathological causes of double-strand breaks in mammalian somatic cells.